all affected cells, it may be only on the breast tissue, or could be in the stomach tissue, endometrial tissue. So those tissue they have a risk to get the cancer. For example, breast cancer are not heritable, occurs in a specific tissue, as I mentioned, a specific tissue, for example, breast tissue, endometrial tissue, and responsible for the majority of the cancer that we mentioned is inherited. So if I ask you in the exam which types is the common, the somatic mutation, 95% are the causes of the inherited cancer, and differences that this is not heritable, affected only on the specific tissue, but the germ actually is inherited cancer, and those offspring also carry this risk to get the cancer. So it's important to do the process of the screening to prevent to get the cancer for that patient and also for the offspring of those patients. So in this table, actually from the Robbins, you see that the inherited predisposition could be autosomal dominant cancer syndrome, could be autosomal recessive cancer syndrome. For autosomal dominant cancer, we have retinoblastoma. The gene responsible for that cancer is RB genes that we are going to discuss regarding that. We have TP53, that's very important tumor suppressor gene, and actually is important because it's associated with so many types of the cancer. For the syndrome, as you will see that Lee-Frommel syndrome, those people that they have this, they are actually they are predisposed to get the cancer for skin, for example, melanoma, squamous cell carcinoma. Another example, maybe we have MEN syndrome. The genes important is MEN1 and RET. For autosomal recessive syndrome, the problem could be defective in the DNA repair, in the process of DNA repair. Example is Gizoderma pigmentosum. These people also they have risk for to get the skin cancer. Fanconi anemia also is another example for autosomal recessive syndrome. Those patients suffering from pancytopenia. Do you know pancytopenia? Pancytopenia means decrease in all three lineage, means as red blood cells, white blood cells, and also platelet. So people coming with pancytopenia means hemoglobin is low, white blood cells leukopenia, thrombocytopenia, and anemia Fanconi is common in the children that they are suffering from aplastic anemia. Aplastic anemia means there is no actually production of three lineage of hematopoietic elements. The problem is a defect in the DNA repair genes, and these people they have a risk to get acute myeloid leukemia as a malignancy of the hematopoietic elements. This is very important. How do you classify the genes that are responsible or actually important for process of the carcinogenesis? So we can classify those genes as growth promoting or oncogenes or proto-oncogenes. So there are four important actually: proto-oncogenes, tumor suppressor genes, regulator of apoptosis. You know the apoptosis is programmed cell death. So if something happens in the process of apoptosis, the cells that they are supposed to die, they are not going to die. So they continue their life and they do proliferation. So there is a risk to get the cancer. So this is another group. The fourth group is those that regulate interaction between the tumor cells and host cells. Remember, last week we said that we have parenchyma and then we have the stroma. The stroma is supposed to provide the blood circulation and blood supply, nutrition for the tumor cells. So we have a genes that regulate the interaction between the tumor cells and the host cells. So this is also important. So we can classify the genes responsible for carcinogenesis as a tumor suppressor genes, oncogenes, those that they regulate apoptosis and interaction between the cells and host cells. So first group. In normal condition, we have genes that they are named as proto-oncogene. Proto-oncogenes, they are normal, okay? And what is the job of these genes? They're supposed to control the cellular proliferation, the cell proliferation. So if they are normal, we have a normal cellular proliferation. If something wrong happens, I mean the proto-oncogenes damage, could be due to viruses we mentioned, could be due to chemical environment. So those affected genes, they are not normal anymore, and we call it oncogene. So you say oncogenes means they are not normal. So when they are not normal, they cannot control the cell proliferation. So the cell proliferation is not under control, and they change the cellular function and proliferation. And since they are not under control, they do process of proliferation. Because there is no control, the cell proliferation is too much, and then what happened? Again, we have process of neoplasia and tumor. So this is a process of oncogenes. So this is another slide. As you see, proto-oncogenes, they are normal cellular genes whose product supposed to promote cell proliferation. But the oncogenes, they are mutated proto-oncogenes, means they are abnormal. Oncogenes, they are abnormal, but proto-oncogenes, they are normal. So when we have oncogenes, means those proto-oncogenes damaged, and now they are not normal, and because they are mutated, because they are overexpressed, they don't have a normal function, and since they don't have the normal function, what we have accelerated cell division, means the cell division or cell proliferation is not under control and is accelerated. Since it's accelerated, we have increase in the number of the cells that they are monoclonal, and as a result, we have neoplasia or tumor. So those oncogenes, they are dominant, means if only one single allele affected, the process of the cellular transformation damage. So it's only one is single allele. So because of that, we call it dominant genes. So if you want to give example for those that important in the process of the oncogenes, could be genes that control the growth factor receptor, signal transduction protein, nuclear regulatory protein, and cell cycle regulator. I'll show you here. See this one? This is, you can consider this is the cell, and from the outside there is a growth factor receptor, and those growth factor receptors, when they receive a message from the outside as a growth factor, they're going to send the message to what? To a cytoplasmic signal transduction protein, and this signal transduction protein going to send the message to the nucleus, and as a result we have transcription factor activation, and we have cell cycle proliferation. So in each step, if something wrong happens, for example, we have a problem in the growth factor receptor, and this receptor is not under the control. So it's going to continue without any message and do the process of signal transduction protein. So you see that signal is coming, and the nucleus also doing the process of proliferation, and as a result we have neoplasia. So this could be as growth factor. So the growth factor stimulate independent expression of the growth factor, and its receptor actually setting up an autocrine loop of cell proliferation. Example is PDGF or platelet-derived growth factor receptor in brain tumor. So if ask you and mean that is it oncogene or is not, and is responsible for which types of tumor is brain tumor, and the name is PDGF. We have a table, as you can see here. These are example of oncogenes that they have the rule as a growth factor. For example, we have a fibroblast growth factor. Overexpression of these genes can cause a stomach cancer. Okay?
Is it important?
Maybe the one, as I mentioned, for example, astrocytoma, PDGF beta chain overexpression can cause brain cancer. The next, remember we have growth factor receptor. Mutation in the genes encoding the growth factor receptor. If something wrong happened in the growth factor receptor genes, now this growth factor receptor is not under control anymore and send the signal automatically without any control. So as a result, again we have cancer or neoplasia. Example is amplification of epidermal growth factor that we name it as EGF, which is a receptor for family of HER2. Is important in the breast cancer. This is very important because we have a specific treatment. If you know the process of the carcinogenesis, we can create a target therapy or target drug for these affected genes. For example, nowadays we have a drug for the HER2, we call it HER13, which is a specific fit for this growth factor receptor and can stop the process of the signal transduction and we can treat the patient by this drug. Actually, what they do, they send for us the tissue. We do the process of immunohistochemistry. We see that the HER2 signal is there or no by immunohistochemistry, and they said, okay, HER2 is positive, so they can start the process of treatment by Herceptin because it's a specific drug for the HER2 receptor. So this is another table. As you see that in this table, there is a list for growth factor receptor in the family of oncogenes. Remember, they are all in the family of the oncogenes. We have EGF receptor family, CSF1 receptor family, PDGF receptor family, and we have different types of cancer, as you can see. The third one, remember, we have growth factor receptor, and the one signal transduction protein. If something wrong happen in the process of those genes that are responsible for signal transduction protein, so now they are not under control, and again we have the process of the cell proliferation automatically without any control. The example could be a GTP binding because of point mutation, KRAS, we have colon cancer, lung cancer. Another example is a BRAF. This one is important. For BRAF, point mutation of the BRAF genes can cause RAS signal transduction, and as a result we have melanoma. ABL, for chronic myeloid leukemia, we have actually translocation of chromosome 9 and 22. As a result of this translocation, there is fusion between the ABL and BCR, BCR-ABL1 fusion can cause this signal transduction protein activation, and we have chronic myeloid leukemia. Remember last week we learned that leukemia is malignancy arising from the bone marrow. Since this one is a chronic, means that there is myeloproliferation.
Chromosome 9 at chromosome what?
22. The chromosome 22, we named it as Philadelphia chromosome. Philadelphia chromosome. There is fusion between the BCR and ABL1 as a result of this translocation. Another group is nuclear regulatory protein. Nuclear regulatory protein, also they are under group of oncogenes. Okay, so if they are not under control and something wrong, let's say mutation, amplification happen or translocation, as a result, again we have activation of these genes, and we have different types of neoplasia. For example, for Burkitt lymphoma is B cell lymphoma, is arising from B lymphocytes. They are mature, they are not blast, they are not, stem cells, they are mature B cells. Now we have translocation. As a result of this translocation, there is fusion of c-MYC and BCL2. BCL2 and c-MYC fusion, c-MYC and IgG fusion, and when we have fusion between the c-MYC and immunoglobulin heavy chain, this c-MYC is very active and there is too much cell proliferation. Maybe today we have a Burkitt lymphoma, the size is like as a lemon, but tomorrow the size will be like watermelon, becomes so big. Its proliferation rate is very high. In each second, maybe 10 times the cells proliferate. So if you don't treat the patient, patient is going to die. But if you treat the patient, patient have a good survival. So for Burkitt lymphoma, also there is translocation. As a result, we have activation of proto-oncogene. The name is c-MYC. And this c-MYC rearrangement is responsible for the Burkitt lymphoma.
IgG and BCL2?
Immunoglobulin heavy chain and c-MYC.
Okay.
Another example is amplification of N-MYC for neuroblastoma or L-MYC for small cell carcinoma of the lung. That's all for the oncogenes. Remember, we had 4 groups of genes. Number 1, oncogenes. Number 2, tumor suppressor genes. Third, those that are responsible for apoptosis. And the fourth, those that are responsible for cell interaction between the host and tumor cells. Now the second group. Second group, the name is tumor suppressor genes. So in the normal condition, they're supposed to control and suppress the proliferation, and they are called as tumor suppressor genes. Okay. So genes that they normally prevent uncontrolled growth, we call it tumor suppressor genes. So they, at the normal situation, supposed to control the cell proliferation. When they mutated or when they lost or transform, so they cannot control, they are not under control, and as a result, there is no tumor suppression. Okay? So as a result, we have a neoplasia. So usually, remember for amplification, for oncogene, we said only one single allele will be enough to get that neoplasia. But here, we need to have both normal allele affected. Means here, those both normal allele supposed to affect it, so it's a difference between the oncogenes. So usually both normal allele of the tumor suppressor genes must be damaged, must be mutated, or must be lost for transformation to occur. So this is one difference between the oncogene and tumor suppressor gene. Tumor suppressor genes, usually they are useful, supposed to control the proliferation of the cells. We have governor and guardians. So example for the governor, which act as an important brakes to the cell proliferation, is the RB genes. How about the guardian? Guardian are responsible for sensing the genomic damage. So the name, an example could be TP53. So if they ask you RB genes is important or TP53 gene, see that the RB is a governor and P53 is guardian. So this is actually protective, but this is actually as a boss, is important brakes.
As what?
This one we see that the RB genes is important brake on the cellular proliferation. The RB in terms of what we call it as the role is more important compared with the TP53 RB genes. But TP53 is sensing damage, is a guardian. So the governor is RB, retinoblastoma. Any defect in RB genes lead to the removing an important brake on cellular proliferation. Okay? So example is retinoblastoma. Retinoblastoma is a tumor arising from retinoblast in the eye, and it's important in the pediatric population. This is name RB genes, is the first discovered tumor suppressor genes. The location is chromosome 13, so this gene is located on chromosome 13. Retinoblastoma is a childhood tumor with inactivation of these genes. Okay, so the group is tumor suppressor gene. When these genes are mutated or lost, there is a chance for the child to get a retinoblastoma. So the retinoblastoma genes, actually the product of these genes is the RB protein. So the RB protein is a product of the RB genes and has a key role in the regulation of cell cycle. So you see this in picture, what we have actually. Here we have a process of transcription between the DNA, and as you can see here, we have RB. This RB we mentioned is the role is what? Is the tumor suppressor genes. So these genes actually produce this RB protein. So how is the mechanism? You see that when the RB, this is RB, when the RB is hypophosphorylated, means there is no, see here this is hyperphosphorylated, too many phosphor around the RB. Here, see that we have phosphor. See that here we have a phosphor around the RB protein. We call it hyperphosphorylated RB. But this one, we have only two phosphors, so this is hypophosphorylated. So when the RB is hypophosphorylated, attached to the E2F. So when the E2F attached to this RB protein, the process of transcription stops. So as a result, there is no progression to space and there is no cellular proliferation. But when the RB is hyperphosphorylated, cannot attach to the E2F, and E2F continue the process of the transcription, activation, and proliferation. And there is a progression of the G1 to space, and then we have a synthesis and cell proliferation. So when the RB become mutated or become actually lost, or something wrong happened in these RB genes, there is no process of the E2F inactivation, and there is a cell proliferation, and this cell proliferation can cause neoplasia. How about TP53? We mentioned TP53 is guardians. Guardians actually is like a protective, sensing a genomic damage, means sensing that there is something wrong in the genes. So how is the process? Actually, these guardians leads to the cessation of proliferation. When something wrong happens, when there is a damage, TP53 sensing that there is a damage. So they try to stop the cell proliferation, giving time to the cells to do the process of the repair. If they can repair, they can continue the cell proliferation. If they cannot repair, they're supposed to die and go under, and they go under apoptosis.
Professor, this slide is not important.
Yeah, it is maybe later on we will explain. We call it Knudsen phenomenon. So the guardian is sensing genomic damage leads to the cessation of proliferation, or if the damage is too great to be repaired, we have induction of apoptosis. So the example for these groups is TP53. We mentioned TP53 is tumor suppressor gene. This is under groups of guardian genes. So TP53 again encodes a protein that we call it P53. Actually, it has a central monitor role in the stress of the cells, which can be activated by anoxia, let's say DNA damage. So activated P53 control the expression and activity of the genes involved in the cell arrest, DNA repair, cellular synthesis, and apoptosis. I give you, maybe I show you a picture here. So it is here. Can you see? We have the cells, normal cells. TP53 is normal. This is a normal condition, and everything is okay. But now there is a DNA damage. For example, exposure to the drug, to the viruses, to the UV, something wrong happened. So there is a DNA damage. What is the role of this P53? P53 sensing this genomic damage and understand that something wrong happened to the DNA. So the P53 try to solve this problem. How is it? They try to fix, they try to repair the DNA. So if they can repair, successful repair, again we have a normal cell proliferation. But for example, the repair is too, they cannot repair it, the repair fails and they cannot do the process of repair. So what we have apoptosis, which is a programmed cell death. So P53 trying to do the process of repair. If they cannot, we have apoptosis. But let's say here we have a DNA damage and P53 also is mutated or something wrong happened in the genes of TP53 as a loss or mutation. So now this P53 cannot understand DNA damage, cannot fix the DNA damage. So since there is no fixation, there is no sensing a DNA damage, as a result there is no DNA repair, there is no process of apoptosis, and what we have a process of cell proliferation which is abnormal in the DNA, so we have a neoplasia. See that here we have a tumor. It's a malignant tumor because P53 couldn't act and cannot show the normal role as a do the process of DNA repair and apoptosis. The third group are those genes that regulate the process of apoptosis. Remember, we mentioned the apoptosis is programmed cell death. I give you example. For example, in the lymph node, if you are familiar with a lymph node histopathology or histology, we have a lymph node as capsule, and we have lymphoid follicles. And inside of the lymphoid follicles, we have germinal center. Have you learned the histology of the lymph node before? So, in the germinal center, we have a lymphoid B cells. This B lymphocyte, when exposed to the antigen, they try to produce the specific antibody with high affinity. So, this process of B cell proliferation happens in the germinal center. And in the germinal center, we have too much apoptosis because when the B cells proliferate, they try to produce a centroblast, centrocyte, and as a result, they have plasma cells, and plasma cells can produce a specific antibody. But when the cells they don't have the high affinity for those antigen. If they cells, these B cells, they are not good, they are not so specific for those antigen, they're supposed to die. We call it apoptosis because of the process of the selection, because they are not so good. Maybe they cannot recognize the self and non-self. Okay, so they're supposed to die. We call it apoptosis. Then we have genes as BCL2. I'm sure that you have learned this one in the cell engine chapter, BCL2. Yeah. And this BCL2 is anti-apoptotic. Alright?
Yeah.
Okay. So there is when BCL2 is anti-apoptotic. Am I right? So when the BCL2 is active, so there is no apoptosis because it's anti-apoptosis. If the BCL2 is become activated, so there is no apoptosis anymore. Am I right?
Yeah.
Okay. So we have a malignancy, we call it follicular lymphoma. Follicular lymphoma. As a result of activation of BCL2. When BCL2 becomes, there is translocation between chromosome 14 and 18. And when these two translocate, the BCL2 become activated. Since BCL2 become activated, there is no apoptosis. And then we have B cell proliferation, they proliferate, and there is no apoptosis. And as a result, we have follicular lymphoma. But at the normal situation, when you see the germinal center, the BCL2 lump is off here. Because of that, we have apoptosis. When you see the normal germinal center, you can see so many tingible body macrophages. Tingible body macrophages, actually what they have inside, they have fragmented nuclei because of the process of apoptosis, because they are a scavenger, because they engulf the result of apoptosis, the choriorexis pycnosis. But when it becomes neoplastic, there is no tingible body macrophages inside the lymphoid follicle because there is no apoptosis, there is no scavenger. So this is example for apoptotic genes. So the anti-apoptotic means that there is no programmed cell death, and the cells they are not under the control, they can continue their life even though they have problem. So as a result, again, we have neoplasia.
Can you repeat the last part about the macrophages?
Macrophages, actually, they are like a scavenger. When there is process of apoptosis, they clean the background. They engulf the apoptotic body. They engulf the result of choriorexis and choriolysis.
They made by a definition of the BCL?
No. I mean, when there is no apoptosis, there is no apoptotic body. So, as a result, you cannot see the tingible body macrophages because there is no apoptotic body to engulf.
Which one is made it and make it?
I mean, the salon. One. So this one, remember we said tumor suppressor genes, we need two allele affected. For first one, we said only one, but here can have both. Could be loss of one copy or could be loss of both copies. So for these genes. The first groups are genes that regulate interaction between the tumor cells and host cells. So these are genes that enhance or inhibit recognition of the tumor cells by the host immune cells. This is very important. Nowadays we have an immunotherapy, and for immunotherapy, the mechanism is by the immune system can recognize the tumor cells, or tumor cells, they have some mechanism that they escape from our immune system. So what we do, we're trying to understand what is the mechanism that the tumor cells escape from our immune system, and they can do process of proliferation without control of our immune system. So nowadays we have a specific drug that they try to understand this process and stop the process of the escape of tumor cells from our immune system, and they control the cell proliferation of the cells. Okay, I think we explained this one before. So the next part of my lecture is regarding what kind of damage happened to the genes that become carcinogens. Could be as a point mutation, could be as a translocation, could be amplification, could be, could be balanced, could be unbalanced. So these are different types of gene lesions. We have a driver mutation, we have a passenger mutation, and driver mutation actually are mutations that alter the function of the cancer genes and thereby directly contribute to the development or progression of the cancer. They are usually acquired. Passenger genes are acquired mutation that they are neutral in the terms of fitness and do not affect cellular behavior but may be associated with clonal expansion. Sometimes you have a subtle genetic alteration, could be as just activation of one protein product or inactivation of protein product. Example for protein product, we mentioned RAS, EGFR, and then inactivation of the protein product like as tumor suppressor genes, as RB gene, P53 genes, they are point mutation. Sometimes we have a karyotype change. It's karyotype change, we mentioned that when there is a karyotype change, these changes are very large and then you can detect it by cytogenetic study or karyotype study. So when you say genetic damage, could be in chromosome, could be in the genes, molecular damage, so could be very large or could be very point mutation. So those that are karyotyping changes, we can study by cytogenetic. Those that they are in the molecules on protein, we need to do molecular study that understand is there any gene or the nowadays we have gene sequencing. Gene sequencing, what they do actually, they sequence the genes and they understand very maybe point mutation or something wrong happening in the genes. So this one we call it genetic study, but genetic study could be karyotyping, could be molecular study could be by FISH, could be by PCR study, and different types of.
RP on TP53 is the point mutation.
P53 is point mutation.
P53 is point mutation. RP is not.
RB also could be as a point mutation.
Okay.
I will show you in detail. So we have the genetic lesion that activate oncogene or inactivated tumor large enough to be detected by karyotype. So this karyotyping could be loss or gain. For example, could be deletion or addition. Sometimes we have a deletion of one chromosome, sometimes we have an addition of entire chromosome or part of the chromosome. So could be as aneuploidy. Some cancers have a normal karyotype. So if, let's say, if you examine the karyotype and karyotype is normal, we cannot say that it's no problem, because it could be as a just molecular damage, could be as a genetic damage, and karyotyping normal. So others are markedly aneuploid. Some cancers, they are very aneuploid. There is a problem in the number of the chromosome, addition, deletion, loss, gain. So by chromosome study, you can understand something happened. This genetic alteration can be non-random and common characteristic of the particular tumor, or could be random genetic damage. We have a non-random genetic alteration, and for example, if it is a non-random genetic alteration, could be a balanced translocation. Balanced translocation can activate proto-oncogene in two ways. For example, we have a translocation, and as a result of this translocation, we have an overexpression of the proto-oncogenes. Proto-oncogenes, remember, we mentioned growth factor receptor, signal transduction protein, nuclear regulation protein. Okay, we have B-cell lymphoma, Burkitt lymphoma. Remember Burkitt lymphoma? There is translocation between chromosome 8 and chromosome 14. So we mentioned when there is translocation, there is juxtaposition of the c-MYC and immunoglobulin heavy chain. Okay? So when c-MYC on chromosome 8 juxtaposed to immunoglobulin heavy chain, there is hyperactivation of the MYC. And as a result of this hyperactivation, we have a Burkitt lymphoma. So this is important. Coming in the exam, most of the time I give you a case scenario. This patient is coming with a lymph node. I give you a case scenario as a child coming with a lymphoma, and they do a study, cytogenetic study, and there's translocation in 8 and 40. Which one is most likely? Also, you should choose it. Another example could be follicular B-cell lymphoma. For follicular B-cell lymphomas, again, we have a translocation between chromosome 14 and 18. And this is a reciprocal translocation. As a result of this reciprocal translocation, we have hyperactivation of the BCL2 that I just now explained for you. When the BCL2 juxtaposed to immunoglobulin heavy chain, the BCL2 become activated, and we mentioned the BCL2 is anti-apoptotic. When there is no apoptosis, there is a chance to get a cancer. This is a picture. As you can see here, we have translocation between 8 and 14. And we mentioned when 8 and 14 translocation, there is a juxtaposition of the semic and immunoglobulin heavy chain. And when the semic rearrangement happens, there is increased expression of the progross genes or proto-oncogene, and as a result, we have cancer. Sometimes there is fusion genes. The example is chronic myeloid leukemia. We mentioned there is translocation between chromosome 9 and 22. Chromosome 22, the name is Philadelphia chromosome. When the chromosome 9 and 22 shows reciprocal translocation, there is fusion between BCR and ABL1. Fusion of BCR and ABL1 cause tyrosine kinase activity, and as a result, we have chronic myeloid leukemia. This is very important because when they do a study by cytogenetic or by molecular and they detect the BCR ABL1 fusion, we have a specific drug for this patient. We have an imatinib, which is a tyrosine kinase inhibitor, and then we can treat the patient, and the patient can survive like as other normal people. So this is important for us if the patient is coming with a leukocytosis to understand that is a chronic myeloid leukemia or not. So what we ask for cytogenetic and molecular to understand is there any translocation between 9 and 22 or BCR-ABL1 fusion, because we have a specific drug for this patient. Another example is a Ewing sarcoma. Remember, we have learned sarcoma is a malignant tumor arising from the mesenchyma. Ewing sarcoma is common in the young patient, children and young patient, younger than 20 years. And there is a fusion between there is a translocation between chromosome 11 and 22, and as a result, we have fusion of the genes, Ewing sarcoma genes and FLI1. When they fuse, there is a risk to get the Ewing sarcoma. Ewing sarcoma, in terms of morphology, the cells, they are small, they are round, they are blue, and they have too many mitotic activity. The cell proliferation is high. And if we don't treat the patient, there is risk of metastasize to the lung and patient can die. So it's important to make it correct diagnosis because there is a specific treatment for those patients as well. This is a picture of chronic myeloid leukemia karyotyping, and you can see here we have chromosome 9 and chromosome 22. And this is the location of PCR locus, and then we have the ABL. And we see that's the Philadelphia chromosome. We have a BCR and ABL1 fusion. As a result of this, we have a Philadelphia chromosome. And because of this fusion, we have tyrosine kinase activity. We mentioned imatinib is a specific drug for tyrosine kinase inhibitor.
Philadelphia and even if both of them have high mitotic activity?
I couldn't understand.
Philadelphia and Ewing, both of them.
Ewing sarcoma?
Yeah. Both of them have high mitotic activity.
Yes. BCR, when these two genes they are fused, they cause the proliferation of the cells. Because the cell proliferation is high, you can see so many mitotic activity. Burkitt lymphoma also has 100% proliferation activity. Means, as I mentioned, the proliferation rate is too high. And maybe today is size is 5 cm, tomorrow is 20 cm because it's too much high proliferation. Okay, so we mentioned translocation, and another group could be a deletion of the chromosome. For example, chromosomal deletion are the second most prevalent karyotyping abnormality in tumor cells, more common in non-hematopoietic solid tumor. Deletion, loss of particular tumor suppressor genes like the RB or P53. Deletion is important for us. Amplification of proto-oncogene may be converted to the oncogene by amplification. So could be overexpression, could be deletion, could be translocation. These are different types of problems. This is a pictures for amplification of the genes, and as a result we have a neuroblastoma. The other group could be aneuploidy. We mentioned we have a gain or loss. So aneuploidy is defined as a number of the chromosome that is not a multiple of the haploid state. For human, that is a chromosome number is not a multiple of 23, so we call it aneuploidy. Occurs due to the error of the mitotic checkpoint. Okay. So the process of the carcinogenesis is a multi-step process. Maybe before this, I show you another slide. Do you need to have a rest?
Yes.
Okay, maybe 5